Okay. Genetics and inheritance, stem cell and cloning. Welcome to my class, guys. Today's date is the 9 April 2024, and the time there it's 7 minutes past 12, right? So welcome to my class, guys. Sanbonani, Dumelani, Avsheni, hello. Right, so I like this DNA thing and the cloning, what the thing that I'm adding into my class, right? So I promise you that next time when you come here, you will find this screen having something at the bottom, life science grade 12, maybe with Mr. Mpepu, or just life science grade 12, right? For those who are selecting these videos for the first time, so that they will, like, the welcoming slide will also welcome them, and they will feel that they are at life science grade 12 and they are not lost, right? So with that being said, welcome, guys. I welcome you and I'm very much appreciated. Did I greet you? Sanbonani, Dumelani, Avsheni, hello, nda. I welcome you, guys. My name is Mr. Mpepu. So last time I checked, guys, we were at 45 subscribers, so we welcome these two. Thank you, guys. Our class is still open. We still have those rows to fill, guys. Another thing, let me appreciate all of you who are subscribing, guys, and all of you who are coming here each and every day, because after uploading my videos I can see my 20 views. Do you understand? So it also shows that you guys are there and are following me. So I appreciate you guys. Lingdramutu. And another thing, I also have a Facebook, I also have a TikTok, all by the name African Transcribers. And then another thing is that we also have a WhatsApp group where learners are 071 190 6168, where learners, they do get some activities, and then after getting those activities, they can share the answers, or if you have a problem you can post it on that WhatsApp group, and then after that those learners who are there, they will help each other, and then the activities will be going on like that, right? So today we are at week 2, Tuesday. We started yesterday. If you are late here, guys, take a seat and relax and enjoy our class, right? Kwabanda, no, you can just put your headphones, get inside the blankets and switch on your phone, and make sure that you set this at a full screen mode, and also press that quality to HD, so that you'll be able to read whatever I'm seeing on my screen, right? So I also thought of incorporating some things. You can go to comment and comment how we can build this class, right? This class is between me and you guys. This class is ours. We are bringing this class to the world, right? So another thing that I was thinking is, since I can go to internet and search a specific name, specific introduction, or a specific information about a specific topic, I will now start to do it here live with you guys. So if maybe like we want to search something, maybe for an example, yesterday we did a genetic engineering, right? So after doing genetic engineering, paternity testing, yes, we talked about it nyana, because you did this term 1, right? And another thing is that we also have genetic links. So maybe for an example, I want to search let's say I want to search a video that maybe suit, or that explain better the process of genetic engineering, the insulin production. I can go to internet and search that video, and we watch it here, right? Maybe what I can do is to switch off the sound and explain it myself, so that the copyright what they won't restrict my videos, right? And another thing, if we can go online here and Google a certain question, and then we find it online, that page there, it will still be there, and we can reference it, right? So that tomorrow no one can come here and say, hey, we used this information without their agreement, without a agreement and that thing, right? They can come and sue you tomorrow. And then if, guys, tomorrow someone comes and sue me, remind me of this video where I'm saying, I'm declaring this, since this is a learning environment, and we are learning, and we are going to incorporate this technology into our learning. So if we find like we want to research something, I can just go straight and research it. For an example, I can do this. I can come to my Chrome here, right? I can come to my Chrome here, and then something like this, and then let's say you see I was searching glyphosate. This glyphosate, the disadvantage of the disadvantage of growth genetically modified organisms, right? So I was teaching that these plants, they come with glyphosate. And what is a glyphosate? Glyphosate is a chemical that they use to, you understand? So if I want this kind of help, now I will come and consult Google. We can see that this is Google. We can see this is Google here, and then the information that I'm sharing with you, you can find it at glyphosate, this HPP oral education / facts sheets / glycogen, right? So if right now you feel like you can go and do a deep what do we call a deep investigation or deep study, you can also take down this what do we call, man, these links here, or you can request them on my YouTube, on my description, on my comments, and then we can come and recopy them and send them to you, right? So this is what I was thinking in this class. So comment on the comment section if you think it's going to work, right? Although it's going to work, it's just that our schedule maybe like I will use someone's video of YouTube, and then that thing, it goes they restrict it, and then you find that my videos on YouTube, they are being restricted, and then they are not being published to reach other learners. So we'll use that, we'll do that. I'm not sure if it's being allowed or what, but we'll use that, right? So we welcome those 43, those two learners that they joined us yesterday. We welcome you guys. Don't forget to subscribe, and don't forget to hit that bell notification, right? So that when my YouTubes are being when my videos are being posted, you will be reminded, like the alarm ya sekolo tlotsena people nyaka go classic, Mr. people are class ya higher ya life science. You come here and then you watch the video. Or also maybe you can say you come back during a night, you come back from school around 5, grade 12, they keep you at school, right? So around 5, take your shower. After 6, and then know that after time when you go to sleep, you can just wear your gowns and what and watch this video, right? And another thing is that I don't know, or I'm not sure, or I can't find a specific place where I can use to see these 45 subscribers, right? So I don't know you guys, I don't know who are you. I just see that my class was added 2 learners, now we are 45, right? So I still wish that we can push this 45 up until maybe 100 before we can mark our 1 month anniversary, right? Because we started at the 16th. If you still remember, it was Saturday. I announced that on the 18th we'll be starting our weeks, and then we started on the 18th. Now we are at week 4. So when it got 18 again, when will be the next 18th, I'm not sure, or 16. So you can also, since this class is for hours, right? Let's do this together. You can comment on a comment section which date can we use as our anniversary date, right? Monthly anniversary, right? So it can be the 16th, the first day when I introduce that by the 18th we're going to start, or it can be the 18th, the first day that we started with these classes. So thank you guys. That 10 minutes was for introduction and was for all the announcement, right? So welcome guys, we're at 45 students, and we also wish to reach more learners, guys. Tell your friends to tell your friends to tell their friends, so that they will tell their friends that, hey, right? So if you need the link, you can also come here, or you can also take the screenshot here. Another thing that we can do, you can also take a screenshot here of subscription, the screenshot here like subscription, and your name there, your what we call your YouTube name, and then you send it to my private account here, WhatsApp, and then say your name, right? Then now I will know that in my register book I have out of those 45 students, I have Mang who subscribed as Mang, right? Then I'll come back here and introduce you to the class. We are also going to choose the class representative. Like, guys, let's do this together, right? Help me to help you to make this dream come true, right? This will be our class, this will be our future class, and this class is going to be okay. Let's move on. Yesterday we did genetic engineering. Welcome to my class, by the way. We are already done week 1, we are already done week 2, we are already done week 3. If you are the first time coming here, you are behind, right? You still have to go back and rewatch these videos, because now we are doing with genetic engineering. Now we are dealing with the real application of this knowledge, right? You understand now that we have a gene, and then this gene is made up, it's cut out from a DNA, right? In this DNA we have a GC nucleotides and nitrogenous bases GCTA, right? So now in this genetic engineering, we are engineering, or we are applying science and technology to engineer, to transform, or to modify a certain gene, right? Why are we doing that? To benefit a human species. You understand? Today we'll be talking about a sheep dolly, the first sheep dolly, and then today we'll be talking about a sperm cell. Let's go to our book. Let's do a revision here yesterday, right? Yesterday we did genetic engineering. I told you that yesterday we have we have a biotechnology, and then in this biotechnology, in this biotechnology we have branches of biotechnology, which include DNA profiling, genetic engineering, and stem cells. They explained well on your examination guideline here. We are still having yesterday's. Okay, so here's the thing, right? They are saying we have a biotechnology, and what is biotechnology? Is the manipulation of biological process to satisfy human needs, right? So we are manipulating, or we are playing, we are playing with genes of a specific organisms, or we are playing with biological processes. With biological processes, these are the processes that are including life form, right? Then to satisfy human needs, we are doing this to produce medicines, we are doing this to produce more fruits, we are doing this to produce more eggs, we are doing this to produce more milk, more meat, more what. So all these reasons that they are pushing us to now manipulate genes, it's for satisfying of a human needs. That's the biotechnology, technology of life. I said it yesterday. This is the technology of life, or it's when we are incorporating science and it's when we are cooperating science and technology. How do you write science? You see, it's science, right? Okay, it's when you are incorporating sciences and technology, and when we say technology, you understand that we are talking about the machines, the robots, the fourth industrial revolution. This is when we are using those technologies to now study life, or to better life, or to make our life satisfactory, to satisfy our human needs, right? So that is what is happening. We are using this technology to better our life, right? We can be doing this for different reasons, but all those reasons, they come up as to satisfy human needs. And then we also said in this biotechnology, since this is the tree, and then we have branches. The branches, we are going to talk about genetic engineering, and then in this genetic engineering, it's when but they say it's an aspect of biological biotechnology, right? And includes this following. And then in this genetic engineering, I said that this is an engineering that is taking place inside a genetic level, right? Then it means that in this thing, we are going to the gene, we are going to be manipulating the gene, we are going to be removing the gene, we are going to be inserting the gene, we are going to be adding the gene, we are going to be subtracting the gene. All in all, we'll be dealing with the thing that we'll be doing, it will be an engineering, where at the genetic level. And then in genetic engineering, we have stem cell research that we'll be talking about today. After that comes after genetic modified organism. What are genetically modified organisms? These are the organisms that consist of a gene that is modified, or that consists of a DNA that is modified. We learned about this yesterday when we were cutting a bacteria's plasmid, and then we added the gene from a human pancreas cells, and then this gene, this total plasmid, we send it back to the bacteria, and then that bacteria manipulates, multiply and do its daily basis while producing the insulin. And that bacteria, now we are going to say that bacteria is a genetically modified organism, and we say that we can use this technology to better this thing by maybe modifying an organism to produce more plants, to produce more fruits, so to say, or vegetables, or we can modify organism to produce more milk. We can we can modify organisms to produce a certain thing, right? So all in all, those are the advantages, and we also have disadvantage. I said today we'll talk about disadvantage, but we are just going to pass by those disadvantages, since at disadvantage we get them. I even talked about them. And the disadvantage is that this process here, it takes time, right? It also takes time to prove whether the thing that we are doing here, is it going to have a side effect in a long run or in a short run, right? For an example, we can say genetically modified organism in a short run benefit or a disadvantage in short term, right? Kori, you might find that these people will find we are allergic. I'm just trying to make up some sentence that you'll understand. Maybe like we are saying someone is allergic to a certain organism. You'll see when we do the advantage and disadvantage, right? And they said in this genetically modified organization, brief outline the process of and the benefit genetic modified. This is a process that we are talking about, Kela process here, DNA recombinant technology, right? You just briefly outline it. Even when you'll be needed to briefly outline it, right? So from there, we are going to talk with a sperm cell research, and then we are going to talk about cloning. So with that being said, we can now move on. Then I can also go to our textbook. This is what we learned yesterday. And then we also learned the process of DNA recombinant here, recombinant DNA technique. And you still remember when I was explaining this process that the DNA is going to be chopped off, or we are going to isolate, or we are going to extract a DNA from the host here, right? So the host, it was the pancreas cell. We are instructing the insulin secreting cell, right? So if you still remember this, it's fine. Today we'll be doing disadvantages and advantages, and then we move to somatic stem cell. So okay, I prepared a small revision here. What is happening with since you are going to do this as your experiment, right? This, they can done this as an experiment level, explaining or showing you a video where an insulin is being produced. What is insulin? Insulin is the chemical, or insulin is this hormone that in mellitus diabetes, you'll learn about this, and they will ask you what is the use of insulin. You understand? Yes, you know that insulin, yesterday you learned that insulin, this insulin can be produced by a genetic engineering. We can genetically engineer a bacteria to produce an insulin. But what is the use of this insulin? This insulin, it's used to treat diabetes, right? Diabetes. I'm not sure if this will be the right spelling. Did I wrote it somewhere? Let me check before I can write things that tomorrow you'll be saying, hey, I remember. Let's leave diabetes, something like that. Diabetes mellitus, right? This diabetes mellitus, they have type 1 and type 2. You learn about this, you know about this, type 1 and type 2. And then we have type 1 where your body can't produce insulin, right? For some reasons, your body now, you find that it cannot produce this insulin. Since this insulin, we produce it naturally. And then this insulin, you know that it goes to the blood and increase the take in of glucose, right? And then you are going to learn this when we do a negative feedback, where the glucose, you ingest a glucose, and then the glucose will spark the increase of glucose into your bloodstream, and this one is going to increase the production of insulin. Insulin will come back and make, and it's sort of like stimulate or make, right? I will use make in this scenario. And then this insulin will make your body cells to absorb glucose, and then this glucose is going to be stored as a glycogen, if I'm not mistaken, right? So another process, when this is you are going to learn this, right? You did learn this about negative feedback in grade 11. You learn about this. So this is what this insulin is very much important. Why are we learning about the insulin? Because this insulin, they use it as a medication to fight diabetes, right? So that your body will be able to reduce a sugar level. Barunali tswekere, right? Tswekere. So the thing, okay, these slides I got them from here, slide share, what for learning purposes, right? So we can move on now. This was the process that I was talking about yesterday. If you still remember, we have step 1, and then in this step 1 we have a bacterium here. I'm not sure if this will be visible. We have a bacterium here. This bacterium, you know that this bacterium is a chromosomal, it's a bacterium chromosome, right? And then we also have a ring-shaped DNA called a plasmid, but a small piece of DNA, right? And it's called plasmid. Then you come, step 1, you come and extract or take out, you come and take out this plasmid from the bacteria. And then after taking it out, you cut it using an enzyme or using a scissor enzyme. This enzyme, they are working as a scissor, right? It's like microscopic scissor. You can use it like that. It's a microscopic scissor. These are chromosomes that they will come and cut this enzyme here. This enzyme are the proteins that they will come and cut this DNA here, right? So and then here's our plasmid. Now it's opened, and then the plasmid cut by enzyme, and then after that you go to another step, this side, the different steps, the another side, where you will go to a human cell and extract the desired gene, right? After taking out the desired gene, you come and insert it here, and then for a human insulin gene, this is in human, the gene that produce insulin is inserted into the plasmid. Then we have the entire plasmid, and then the next step, we take that plasmid and inject it inside the other or that bacteria that we remove, or any other bacteria, right? And then engineered plasmid inserted, and then this bacteria now becomes a genetically modified organism, and then this organism will undergo the day-by-day process where it will multiply, and then after multiplying, protein synthesis, DNA replication, and then while that that bacteria is multiplying, it's also sharing that gene here, insulin. So we end up having a huge number of bacteria producing this insulin. Then step number 5, we cultivate this. To cultivate is to take out, right? So this is the process in a nutshell. So we can go and now talk about it. 30 minutes time break. 30 minutes before the stem cell, right? And then step 1 is the preparation. Step 1 will prepare here they were talking about this process. They were using the process to isolate mRNA, right? But all in all, we can just come and write that the DNA or the gene, specific gene from a human being or a human DNA is going to be extracted in step number 1, right? And step number 2, you are going to cut the plasmid with a restricting enzymes here, restricting enzymes. These restriction enzymes, they're saying they cut at a specific recognition nucleotide. So it means that it will come and cut specifically here, and then they will leave something like this. They will leave something like this side, and then that side will also have something like this. And then when those chromosomes come, and then you will see here, right? And then they put this thing is called the sticky ends. We are just passing by here, right? I just want to keep time and have time for stem cell class. Then another thing here, after that, the third step is to recombine. We are say combining. We take the gene here. You can see the insulin gene here. It's now being combined. You can see that thing that I was trying to draw, right? It does not come and cut it straight like this, like guanine and cytosine coming and being cut like this. No, it will leave something, maybe like say G, and then C, G. Then it will come and cut here, right? It will come and cut here, right? So another cut is going to be like, as you can see, another cut is going to be like this, so that when this G and C meet, these two, they will join perfectly, right? So the first step, you take the recombined plasmid or the recombined DNA and transfer it back to the E. coli, which is the host. They can they can ask you the bacteria that is used for recombinant plasmid. It's an E. coli, right? We also have other bacteria, and we also have other processes that we are adding to plasmid using this method, right? It's not only for insulin production, and it's not only the E. coli that it's used. We can also use the yeast, so forth, right? But the same thing is happening. We take out the DNA, cut it, insert the new one, transfer it back to the bacteria, and then that bacteria will do its day-by-day processes up until it's divided, and while it's dividing, it's reproducing that gene. So we move on, right? Then we go to the production, and then this production here. After that, the cell needs the nutrition in order to divide and live. While they live, the bacteria cell turn on the gene of a human insulin, and the insulin is produced in the cell. When the bacteria cell produced by dividing the human insulin gene, it's also produced in a newly created cell. That statement is straightforward. And then here's the summary of this process, right? We have a human cell here, and then we extract a DNA. If you still remember at the top, they were talking about mRNA, right? So this one, there's also another process where you also have to go back and decode this mRNA. Let's say, for example, we have UUA, right? So it's going to decode it and become adenine thymine, and then we are going to match these two, this strand with another strand, and then this here is going to be thymine adenine. So these two strands now will become our DNA. So this DNA now will come and rejoin it here. So if you start with mRNA, there's a lot of I'm just saying that it's possible to start with mRNA, but you'll still have to decode it back to the DNA strands, because the plasmid here, it's a double DNA strand. So this mRNA cannot become and joined with this DNA as it is. I hope you understand that, right? But at the top there, they use mRNA instruction. Then after that, with human cell, you remove the DNA. After removing the DNA, you cut it using a specialized enzyme called restriction enzymes, right? You don't have to know the name, just know that these are the restriction enzyme, or you can just say enzyme, it will be fine, right? And then this is an insulin gene, and then you have a restriction enzyme now cutting that plasmid removed from a bacterial cell, right? You remove the bacterial cell, you have the DNA here. This will be the chromosomal DNA, this one will be plasmid DNA, right? DNA double helix, then we'll cut it here. After cutting here, we are going to recombine it with this insulin gene here, and then now we'll have a recombinant DNA. Means that the DNA, they've recombined, and then we transfer that to the bacterial cell, and then that bacterial cell, as you can see here, it's having those two genes, and then this bacteria will divide. You understand? It means that production will have to provide this bacteria with what we call them optimal conditions, right, for these bacteria to develop well. We also have to produce to provide nutrients and all those things that they will influence this bacteria to live and survive. While doing that, it will be dividing and dividing. While dividing, DNA replication, this double strand of us is going to be replicated into an exact copy, right? Something like this. So this is what is happening with recombinant DNA. So today, and then we have reference there for those who still want to study further about this process of DNA replication, DNA recombinant technology. And then thank you for listening. But any questions, right, I will listen to the questions on the table on the comment section. And then we also have advantages and disadvantages of these plants or these animals that are being produced. Our book here is straightforward. Advantages of genetically modified plants, right? These are for the plants, and even for the animals can be like this, but because the more animals are not being involved in this, right? That's what they are saying, but we are not sure. They will come mostly and say name 2 advantage of genetically modified plants, or name 1 advantage of a genetically modified organism, right? So these plants that we are going to produce by adding one plant with another plant to produce another desired plant, they are saying that they have pest resistance. This is an advantage. They are pest resistant. These plants no longer taste good to insect. It means that they are not going to be eaten, right? We have your flower here, and then this flower here, advantage Kuri. When we recombine a DNA with another DNA, we can produce plants that they are pest resistant, that they taste bad. They don't taste good in insects, which means that these insects, they are not going to feed on our plants. That's an advantage, right? And then another advantage is they say that these plants, they are herbicide tolerant. The crop plant is immune to poison, so large amount can be used to kill the weed, right? This herbicide tolerance, herbicide is the chemical that we use to kill weed, right? Weed here are patch. reward the small. If you are planting a leaf, a maize meal like this mealy meal, you have a mealy meal here, you know that sometimes you have the red small crops that are growing around the mealy meal. We have to go and plow those things, remove those things now and then, so that our plant will grow big, right? But in a huge farming, those farm that we see when we go to Free State and what, those things, they grow, but how do they control them? How do they kill those weed that they are growing inside between this thing? They don't go around there and be plowing using our natural method, right? They use chemicals, and then this chemical, they are called herbicide. When they use this chemical, they are going to spread the chemical, and then after spraying the chemical, this chemical is going to kill these small plants that is disturbing the flow of nutrition to this mealy meal, right? And then because this mealy meal here, they are genetically modified, it means that this herbicide, they are not going to affect them, they are not going to kill them. Here they say the crop plant is immune to poison, so the large amount can be used to kill weeds. It means that because these plants, they are immune, they are not going to be affected by this poison. We can use more this herbicide to kill more, or to kill different types of small plants that are growing down there. You understand? This can also act as a disadvantage. What I was googling before, this can also work as a disadvantage that side, because these plants now, it means that our mealy meal, it's coming with these chemicals here, this herbicide, right? Although they are tolerant, it means that this chemical is not killing our plants that we are planting, but the chemical is going to be transferred to to the mealy meal that we have, and this mealy meal is going to be introduced to our human being. Hence you find that they'll say, hey, wash the fruits before you eat, hey, wash what before you eat, because this these plants of ours, or this fruits that we buy from big companies, they are using herbicide, they are using these chemicals to kill, or to, you understand that I'm not sure what that process is called, utawula ngachivenda, right? Ngachivenda zvividzo pfi utawula, ichitsonga andisitivi kuri vasvivita kuri yini, in Zulu I don't know, angazi kuti siyenzani, but siyayenza lento. Even the Sutus, we do this thing yauri, when we plow our mealy meal, we still go down there with our whatever the material that we'll be using. Maybe you'll be using a fork, right, to remove those small weeds. We call them weeds, right? We call them weeds. We remove those small plants there. But into the large farming, this is what is happening. And there's a disadvantage also. Advantage, also a disadvantage. Because this herbicide here, they will be introduced to the fruits, and then now we'll have to ingest them. And that phosphate, glyphosate, that I was talking about, we get it from here, right? And then you can only just have to know that you also have to only understand that these plants of ours, advantage is that they are disease resistant. Like you see that our plants at home, they will be affected by something. You might find that maybe there is a you understand, a modification. Those plants now, they are weak, they are being infected by some worms or some what, right? So because they are genetically modified, they become resistant to diseases. They don't get sick easily. You can just write that. Advantage is that our plants, they don't get sick easily, but these plants are hardly and do not get affected by diseases, right? And then another point is that it improves food quality. We talked about it, that if yesterday I have a miller that produce small but many many maize meal, right? Mealy meal, right? And then we also have another mealy meal here that produce big mbilas, or big chikori, ne? Sfaki, produce big millers, right? So it means that we can add gene from this, and with gene in this, and we have we have a tall mealy meal that produce large mealy meals, and then right here more, here large. Increase the two, we have more large fruits, right? So it improves the food quality, but do not have damage due to pest and disease, so it looks good. It also makes that these pets, because they are genetically modified, these fruits that we have here, they now going to be pest resistance. They are not going to be affected by pests. They are not going to be affected by insects. It means that they are not going to be affected by diseases, so they will look good. Right, that's another advantage of genetically modified plants. And then going forward, they say they have a cold tolerant. Right, so this is what you are learning. Also, the cold tolerant, rice and tobacco have been engineered to be unaffected by sudden drop in temperature. So they also have cold temperature. We can also include this together, this and this together, because here they say there's a drought and salinity tolerant. So this thing, we can say these plants of ours, they can grow in a cold temperatures and they can grow in an unsuitable environment, right? So this is what they're saying. This helps plants to grow in areas previously unsuitable for agriculture. Drought, it's when there is no water in that place, and then salinity, it's when the place is salty. Right, so we have places where you'll understand, even in a place where there's salt deposit, we also have another organisms that are growing there, or we can genetically modify another organism to go and survive in a salinity environment or in a drought environment. And it's also in a cold tolerance, also in a high temperatures, like venda. Like, do you understand? You understand that we have a venda side, where in venda side we have a high temperatures. So in those high temperatures, we can genetically modify our plants to survive there. These are the advantage. Another advantage is that we can genetically modify our plants to survive at a drought environment where there is no rain. Also salinity environment, also the coldness, where Lesotho side where there is snow. We can genetically modify our plants so that they can also survive at a cold tolerance. Maybe we go there at Lesotho, I'm using Lesotho as an example. We go at Lesotho and we find some plant that is surviving at those snow environment, right? Then we take it gene and we come to Venda and we take avocado DNA and we mix these two and we have avocado tree that can survive at Lesotho. So we end up now having avocados that are being planted in Free State, sorry to say, in Free State. Right, can even ship them to Lesotho. So even a drought and salinity environment, these genetic modified plants can be genetically modified to survive there. I hope you understand here. I hope you understand what these advantages are. Right, because we're now understanding these advantages, it have to spark that interest of you being a genetically modifier. Right, now coming out and say, okay, here in my environment, let's take an example. Here in my environment, temperature changes, right? We have cold temperatures, we have hot temperatures. So I'll have to have plants that they can survive with these temperature changes. So it means that I can go and genetically take something, even if we are not saying genetically going inside the tree and remove and extract the DNA like what we were doing with DNA recombinant. You can just go and take a banana leaf. This thing, you cut a tree, right? You go sorry, I want to say a mango. Let's use mango because banana is something else. Mango tree, right? So you can go to the branch and cut that where am I getting this banana? That mango branch, right? And take it to, we also have mangoes in Free State. So it means that mango in Free State, in its DNA, it's containing something that for survival of it at that condition. And mangoes in Limpopo, they contain a gene that makes them to survive into the harsh environment, hot temperatures, over 40 degrees. So we cut the branch and we merge them, right? After merging them, we have a tree that can survive in both environment, right? And maybe like with this side, we take the one that produce large mangoes, and then this side we take the one that produce more mangoes, and then we merge them together, and then we take this tree and we sell it to Lesotho, even there in England and what. Right? This is what we can do. And then another advantage is that they say that nutritionally enhanced. With this DNA, what we can insert or we can add more vitamin A to rice. So we can add nutrients. Yeah, sorry to say nutrients, and then we say nutritionally enhanced. We did nutrients in grade 11, right? So we can add them in using this DNA genetically modification. We can add this vitamin, right? So another advantage is that we can incorporate vaccine into bananas and potatoes. Maybe the bananas was coming because of this banana here. Incorporating vaccine into bananas and potatoes. This means that the vaccine are made up of plants which can then be transported to countries easily without having to be refrigerated. You know, Uri, when we produce vaccines, we have to refrigerate them, right? We were vaccinated not so long, yeah, COVID, right? So you'll remember that that vaccine they took out from from ISIS, from ice, right? ISIS from ice. Then they take it and then they inject it to you. There was a reason for those vaccines to be placed inside ice, right? You did this when you were doing grade 11, when you're doing what we call this immunization, right? So immunization is also part of biotechnology. So you'll learn that this vaccine they should be in an ice. So for other process to produce this vaccine, we can take the gene. Let me tell you here what we can do here. Here we can go to a, maybe like let's say for example a bacteria, and take out the gene, and take that gene and incorporate or add it into a banana. And then now that banana, they are going to be reproduce the bananas that in their DNA we have we have what? We have this D gene, right? So it means that that banana, when that banana reproduce or replicate, that cells, they are going to reproduce those proteins that we added here, right? So that protein, they will be inside that banana there. So now we can go with that banana that inside it's reproducing, and then inside that banana, while that banana is reproducing, it's reproducing or it's producing a specific protein. When that specific protein, we take it out, we can use it as a vaccine or we can use it as another medication. So we can smuggle using this banana. You can smuggle using potatoes. You just take a potato, you insert a certain gene, you send a message. So you see, even the communication that they are not going to decode, right? If you work about this conspiracy theory, sorry to involve it in this class, but we can communicate in a way that other people or other religions or other terrestrial information like the aliens and what they won't be able to decode our messages. Imagine giving you a potato, taking potato here, taking it to Zimbabwe or to Zambia or wherever there in Rwanda. And then in Rwanda, we have a class or we have a lab that they can go inside that potato and cut it, remove a cell. After removing the cell, they put out they stretch or they place a DNA in an order, and then they go to a specific gene. They use enzyme to cut that specific gene. After cutting that specific gene, they have a mRNA. After mRNA, they add the amino acid. After adding the amino acid, now they have a protein. Now we know that that protein, it has a specific use into the environment or into the people. Maybe now this is a vaccine for something. You understand? Or a message it's on its own. When having that DNA gene, having that GCGAA, GCCCGAA, you know that this is a secret code. When we have a code of GCGCGCCCT, we know that these three and these three and these three, maybe these, they go for the name I am, maybe like I am going there. Right? Maybe this is a message that is being incorporated in this. So now you can send them. Let's doing that. This is another topic you can comment on the comment section, guys. This is another topic that we can talk about in this, right? I also have to do stem cell. Okay, it's fine. And then let's go to the disadvantage of genetically modified organism, right? The genetically modified organism also have a disadvantage. It's costly, but GMO contain glyphosate due to expensive spraying of herbicide, I told you, and then they are expensive, so only rich countries can benefit from this. For you to remove the DNA and what it's very expensive, right? They are saying that we are not sure because we don't even have a public laboratory where we can just go there and do these experiments and see if the DNA can be used to do something else, right? And then another thing, They said the possibility of error is great as the process is complex. You can find that you are adding a certain gene to a potato gene, and then when that gene code with another gene, they produce a protein, and that protein becomes acid, or that protein become deadly, right? Or if maybe you are using microorganisms, now you produce an organism that like COVID-19, right? A COVID-19, a structure of COVID-19 is the same structure or something similar with HIV. So now you can now come here and say, what if that COVID-19, it was a process of genetic modification that went wrong? Because here they said a new pathogens could be made for a biological warfare. So it means that we can make a pathogens, or we can make, you know what is pathogens. These are the or microorganisms that induce your body understand, they cause diseases. Yeah, these are the organisms that cause diseases. So we can produce a new organism that we can go with war, like what Ebola, right? Don't want to talk much here. What Ebola, you can find that Ebola was produced somewhere inside a laboratory using a recombinant DNA, and then that Ebola was taken and then spread in Africa. You same as HIV, right? Same as COVID-19. No, so we are not sure. But with this information, that's the critical thinking that you have to imply on your brains. With your understanding of life science, that are the critical information that you should sit down and say, I've learned this with Mr. Paper. Where can I use this information? Ne? So let's finish this. And then they said there has been a long-term safety testing as a relative new technology. Because this is a new technology, we haven't tested it in a long term. So it means that we might find that we are doing this DNA thing, and then a new organism that are being produced, and they are multiplying, and then it's just a matter of time that this organism, they are going to burst and spread the whole world, right? So we are not sure. We are just tampering with a human, a God creation that one day it can backfire. Hence, there they say that ethically there is a fine line between what can be done and what should be done. So here they're saying that what can be done, maybe what can we do, what we can do, and what it should be do. Like for an example, you can incorporate this gene, or you can cut gene of a human cell with this recombinant what. But what are you? You understand the consequences and what. So hence they say some religious people, they will go against this and say that whatever you are doing, since you are tampering with God's creation, whatever you're doing is not allowed, right? So now you are going to learn about cloning, this thing, stem cell research, right? Then you'll get to understand where these people were coming up with when they said when they say what, when they say that this is ethically wrong, right? And then we have people may be allergic. I talked about this. And then widespread of GMO may lead to loss of diversity. So you might find that with widespread use of genetically modified crops, you might find that, okay, now we have plants that they are resistant to certain diseases, and then that plants, they are going to cross pollinate, or they can pollinate with other, and then now we end up having one type of plants in the whole world, right? You don't find diversity in those plants thing. You don't find those red plants that you find down there because they were killed or something like that, right? So but only time will tell whether the GMOs will solve the food security issues. Theoretically, the potential benefits are huge, but and they say theoretically here, but there are significant risks. This also applies to the medical field and the development of new drugs or ways to administer the drugs, right? The way to administer the drugs, the way to introduce a drug to a human being. So hence, theoretically, the potential benefits are there. You'll agree with me, right? That theoretically, the potential benefits are there. We can produce different things. We can have a vast of food security. We can produce huge watermelons. You understand theoretically, right? But there are significant risks. When you don't know, when you match this, when you take this gene and mix it with gene, what is going to happen? What if you are producing something that is going to be deadly? But you can play it safe. We have animals that we can test these things too. We have rats, we have what that after we produce a certain drug, we apply it to that rat, and if that rat survive, it means that human being will survive. But if that rat dies, it means that thing is toxic to rats. So, okay, with that being said, thank you for being here. Thank you for listening, guys. Thank you for wasting your 50 minutes. And we can have a 5 minutes break, guys. And then after here, we can now go to a stem cell. Because stem cell, we have to do it today. I just wasted my time, or I just felt like I should explain the advantage and disadvantage thoroughly so that you'll understand them. Because I understand that some of the teachers, they're just going to come here and brush this topic and move on like that, right? You know, the disadvantage is expensive, and there's a possibility of errors. Then they move on, because you only just have to maybe to know 2 or 3, or they may ask you, give us a short-term disadvantage and a long-term disadvantage. A short-term disadvantage is a disadvantage that will happen in a short period of time. Example here, we'll use this one, the allergic one, right? People may be allergic. This one will have to happen in a short period of time. When as a human being, just come and eat the nut, and then that nut makes you allergic. So, and long term, what will be the long term? The long term, it will be this one, that after a long period of time, we will lose diversity, biodiversity, the diversity of life, like to have different species and different kind of animals or of plants, right? So in animals, you know that to these days, in chickens, right? Chickens that we are breeding today are those that for eggs and those for meat, white one and the cockroach one, right? Then you know that in that, those are maybe genetically modified chickens, and then you end up finding that those chicken now, they are the most that are being bred into our homes. We no longer have those jacaranda chickens, those chickens that they have a chicken that they don't have feathers here on their neck, right? You still remember that? So yeah, that will be the long term. They will ask you, what are the long term of a disadvantage of genetically modified. They want something that it will happen after a long run, right? And then another one, you can say a short term, this glyphosate is toxic to a human being, so you can digest it, or it can be toxic to you. Then another expensive, and then another short term, maybe like the expensive one, and then this one, they are not safely testing, right? So for them not safely testing, this will affect us in a long term, right? You might find that we are producing cancerous you understand, maybe like you produce an apple via genetic modification, and then now I come and ingest that apple, and that DNA goes to my those amino acids inside the protein. They break down into nucleotides. Those nucleotides, or those amino acids, they join up and form a specific protein. That protein is going to disturb the mitosis of one cell, and then that cell now start to divide what. So that's the process of cancer. But I was explaining how cancer is formed, right? Uncontrollable division of cell. When a cell is dividing uncontrollably, we say now that person is a cancer. Hence you find that some of you do you have the thing here on the ear. You have a thing here on the ear, something like maybe after piecing in your ear, right? This thing developed here. Then they will say, hey, cancer. It's only a name. They have a name of this cancer. But what is happening is that the cell inside, they are now just dividing. You understand? The division now is just happening randomly. It's not organized. You know that the cell has to undergo 4-step interphase, metaphase, anaphase. And this interface, we should have some processes that will happen, and that will trigger the DNA replication. Then after that DNA replication, something will now be triggered for this cell to undergo mitosis. So in this state here, the process of mitosis is just happening random. It's just happening rapidly, right? The cells are no longer regulated. The system that are there in place to check that this system of mitosis is happening at right time. It's not happening, right? So comment, guys, on the comment section. Subscribe. Don't forget to subscribe. We are still moving on, guys. We are just taking a break now. 35 minutes break. We are coming back, right? So that was it from disadvantage and disadvantage. It means that today we might end up 30 minutes. So because I still have to talk about cloning, right? And then you guys will tell me with this cloning, do you think that, here's the question, do you think that the cloning was true or false, right? Do you think like it's a story, right? This thing of if you don't know this thing of cloning, I'm going to explain it today. I'm going to explain it, right? I'm going to explain it. But if after me explaining this process of cloning, or if you know what is happening with cloning, do you think that this process was successful, or they just came up with a new story now and said the cloning was perfect? I'm not sure, because if it was like that, we were now going to see, I'm not sure. But guys, you'll see what I'm talking about when I explain it on a stem cell. You will see what I'm talking about, if that is possible. After learning this life science, I will take it as you will learn life science from a grade 10 up until now. Grade 12, you're taking this into a deep explanation of things. So I want to ask you, with your understanding that a sperm cell with 23 chromosomes will merge with a human egg with 23 chromosomes, and this thing is going to called a blastocyte, and this blastocyte is going to embed into the endometrium, right? This is the process that this sperm and egg will have to undergo, called fertilization. And then while fertilization, this is what this thing this is where this thing cloning comes and puzzle me, because we understand that we have an egg, right? And we have a sperm cell, and then in that sperm cell we are only going to take the 23 chromosome with whatever it was there inside this head, right? And then the mitochondrial DNA is going to be left outside, and then the acrosome there is going to be for some. This is the knowledge that we get from this life science. I'm not talking something else from somewhere, right? And then this acrosome is going here, it's coming here, and it will it will dissolve this layer, and then this 23 chromosome, whatever that is inside this cytoplasm of this cell. Again, this is a cell, this is a cytoplasm, going to be ingested inside here, and then this chromosome, this egg now is going to have a nucleus here, a mother, and a nucleus of a father. So what is happening after here? This cell, this is a one cell, right? It's one cell having two nucleus. So what is going to happen? This nucleus, they are going to dissolve, preparing for mitochondria, metaphase, anaphase, and interphase, metaphase, anaphase, right? This is what they told us. If I'm lying, you will tell me. This nucleus is going to dissolve, right, after the DNA, after these chromosomes they have replicated, right? So after that, these chromosomes, all of them, that means that now we will have 46 chromosomes, 23 from this and 23 this. They are going to align at the equator, all of them, right? So this cell is going to be like this. So what is going to happen is that this cell here, these chromosomes, they will be pulled to one side, and this chromosome, they will be pulled on one side. This is a one cell inside something called an egg. This is happening inside an egg. So this is what is happening. This cell division, this mitosis, it's happening inside something. Because when we have a blastocyte, we have the cells that surround a blastocyte, and then you understand. So here's the thing. This cell is dividing as a one. Sorry to say, this cell is dividing on the one. I was trying to come up with the explanation, and I'm trying to explain to you what is going to happen. So this cell here is going to divide, right? This cell is a one cell. It has a layer, man. If it was not going to have a layer, these cells, after division, a cell is going to go another side, and then another cell was going to go its own way. But we end up having a layer, this layer that an egg layer. Yeah, that's what is going to happen. This egg layer here, it's going to be like that. So after here, these chromosomes, they're going to be pulled one aside. So we'll end up with two cells inside this thing. These are the two cells inside this thing, and this cell here, it's having 43 chromosome, and this cell here, it's having 46, man, 46 for it to say 46 chromosome and 46 chromosome. And this cell here, they're going to divide like this now with this 46 chromosome up until they form something called a blastocyte. And so here's my argument. The cell here in a cell, the cell here in a sperm cell, it's bringing 43 chromosomes, right? These 43 chromosomes, they are going to, before this, this is the process of Let's go now and explain what is going to happen with cloning so that you will understand what I was talking about here, right? Our break is fine, it's done. Thank you, welcome back. That was our 5 minutes break, guys. Today we'll push this maybe up until 1:30, but the stem cell in this book is straightforward. Why I want to add, I'll just add because of I want to share as much education with you, but as limited as a grade 12 student, right? The information has to be limited, but the information has to be there. You have to learn something, but in a limited form, like the types of enzymes. You don't have to know them to understand. Even here in cloning, you just have to know the process of how this cloning was happened and what is this cloning. So okay, stem cell technology, another type of biotechnology, right? But in stem cells are undifferentiated cells, right? Now let's learn about stem cells. What are stem cells? When we say a stem cell, what is it that we are referring to? We are referring to undivided cells, right? That have the ability to grow into any tissue in the body. So it means that these cells, what I will tell you, example, you'll see an example of where do we find these cells. These cells, when they say they have been undifferentiated, a cell when after mitosis, right, I will take it from there. The cell after mitosis, that cell when it's now building back its nucleus and what and producing everything that will be needed for that cell to activate. That cell is still called, or it undergoes a step called a stem cell. This cell it hasn't differentiated. If this cell is like this, it means that this cell it hasn't become a skin cell. I'll use those tissues. Yes, let's use tissues. You did tissues grade 11, right? We have a tissue, different tissues. We have skin tissue, we have a liver tissue, we have a heart tissue. You'll understand that all these organs that I'm mentioning here, they have different tissues, right? And then in these tissues, it's composed of a different cell, right? You find that in the muscle, we have those long cells like this. You understand? Different from the cells that they made up a heart. Maybe like they are you Kerenkaima, what you know this, you did these cells in grade 10, right? If I'm not mistaken. So a stem cell, it's a cell that it hasn't decided whether it wants to come a muscle cell or it wants to come a liver cell, right? So it means that everywhere in your body you will find the stem cells, because everywhere in your body those cells, they are undergoing mitosis, they are dividing, right? So here's the thing, with stem cell technology, we are going to take these stem cells, these cells that they haven't decided whether they want to be a muscle or they want to be a liver, right? And let's take, maybe we take them somewhere. Where do we take them? You'll see where we take them alongside here. And then yes, they say embryonic stem cell and the adult stem cell. I will explain that, right? So we take this stem cell and insect or inject them into muscles like this, right? So these cells, they are, when they are when they are around this, so it's the environment that triggers them to change to that specific cell, right? So it means that if I take this stem cell here and inject them into the muscle, these cells, because the chemicals and what that are composing these cells here, is going to trigger this stem cell of ours to be a muscle cell, BBL. Marawali now. This is what they are doing. They will go to I'm not sure if this is what they are doing. I'm not sure. I don't want to lie, but we can explain it in that way. We see them going to the cells here into your stomach, right? And if you don't know the process, go and learn about it. And then after that, they come to your buttocks and then they inject those cells. I'm not sure if that works, but if this is processed, we can also use this process in this man. Then when these cells here come to your buttocks and now they see, or the environment at your buttocks, it's influencing the stem cells to become the buttocks. It means that your buttocks is going to increase in a shape. Now you find that your buttocks is increasing, right? So let's come and learn. This is what they're saying. This is what are stem cells, right? The stem cells are undifferentiated cell. What are different? It means that after differentiated, they are now become a a muscle. So what is a muscle cell? A muscle cell, it's a differentiated cell. What is a liver cell? A liver cell, it is a differentiated cell. So we can find this cell all over the body, but we have a specific place where we find them. Right, we can go to a specific place and instruct or remove those stem cells. Where and where? But the ability to grow, you see, that they have the ability to grow into any tissue in the body. Right, that's what I was explaining. So thank you. And then they say they may be harvested from embryos left over after the HAIV treatment from bone marrow, and then from bone marrow again, this is another source, and from blood in the umbilical cord. You can also collect the blood from umbilical cord, and then skin. They also say skin and cartilage stem cell may also be used. So we can also, you see, they can also be used. It means that this stem cell, they're everywhere. And then we can also use the skin and the cartilage cells, right? Stem cells. We can also use the umbilical cord stem cell. We can use a bone marrow stem cell, or we can use the embryos that left after. Here they are just putting this information so that people, they don't get angry. But this is what is happening in AVR. What is AVR? Right? AVR is a process where we go and remove a sperm cell, maybe with a needle. We remove a sperm cell, and then we go to the egg, and then we insect the sperm cell. Okay, we go to the sperm cell, we remove the nucleus of it, we remove the DNA. I agree, you know that in a sperm cell, we only need a head. The chromosome white part, we don't want it, right? So we can come and cut this chromosome and the tail side, and then we take this, maybe we poke the acrosome, and then we take whatever it's inside there and pour it inside the egg. And then in that manner, we have successfully fertilized the egg. Now our egg is having two nucleus and the composition that this cell bring. I get this cell, when it bring that 23 chromosome, they are not just drying. Like, they are not just bringing a stick. They bring with chemicals inside. So it means that we have some micro thing that maybe we have what in a cell. What do we have in a cell, in a human being cell? We have Golgi apparatus, we have ribosomes, we have mitochondria. We'll find it in a human. We have centrosome, right? Yeah. We also, in this when you learn fertilization, you were told that a sperm cell, an egg, does not contain something called centrosomes. They're the centrosomes, right? Those things that they produce the spindle fibers, right? We were told that it is not found in an ovum. It comes with a sperm. You get me? So this sperm, these 23 chromosomes inside this sperm, these chromosomes, they also have something, or this head here, it also have something, with the ribosome, with these centromeres, that when we infuse them, so we'll need the whole head, chela, inside the egg, right? We don't take the DNA only, because if we can go just and extract the nucleus only, we are going to lose other particles or other properties that function to fertilize an egg. So that is where I want you to explain this chromosome what, this cloning what, right? So okay, this AVF, I told you, intravial what, fertilization what. So we take a needle. After taking a needle, we remove the sperm, take the sperm, insert it to the egg, and then we artificially. You learn about the word artificially yesterday, right? We artificially. It means that if two people, they don't, if maybe you have a sperm, and your sperm, they are not being able to break open their production, maybe there's a mutation and they are not produced with an acrosome. So it means that they won't be able to penetrate the egg, right? So this process, that's where it can become handy. We come and we remove that head of your sperm, and we insert it straight to the ovum, AVF. Then we take this ovum, we insert it back to the human being, that human being, she's undergoing ovulation, so that process of mutation and and the preparation of endometrium will be ready. The endometrium of it will be ready. Then we insert that egg inside the fallopian tube, and then that fallopian tube, that egg, it undergoes the processes that it undergoes in a fallopian tube up until it reaches the endometrium, and then the implantation will take place. When that implantation take place, then that person she's pregnant, and that person, we call that person a host or surrogate, right? You'll hey, she's a mother, she's a surrogate, she's carrying ngwana wa mang mang, right? We call that person a surrogate. So let's move, right? And then from there they said we can get these stem cells, these undifferentiated cells, we can get them from an embryos where in embryos. Okay, this egg here, after getting inside the fallopian tube, right, it's going to metastatically divide. Mitosis, right? Mitosis is going to happen. Still, when we were singing umitosis, zygote wangena ku mitosis, mitosis wasniki morula. Then you'll see the morula is like this, right? And then this morula undergoes mitosis and it gives us a blastocyte. And as you can see, we have a layer that protects whatever that we are having here. We have the blastocytes. I'm not sure if these are also a cells. This also, they said they call tripo that when these cells they attach with the endometrium here, they now stage themselves and then produce a cilia, villi, what the thing, right? So these blastocytes here, these are the embryos that we know that these cells here, that before the implantation, the cells are undifferentiated cells. We have the stem cell. Hence we have these type of stem cells. The embryo stem cells, they work better because they can differentiate into a different part of the body. Because these cells here, they form a fetus. They form a full body. One can form an arm depending on which side of a fetus that cell is at. If you are saying that this is a blastocyte and this is an implantation, and then we are going to have these cells like this, and then we also have these cells like this, and this cell here is in this side, so it means that this cell is going to produce an arm, and that cell on the other side is going to produce an arm, and the other side is going to produce leg, and that side is going to produce egg, and that side is going to produce. So if we can come and take these cells before they can be influenced to produce something, we can use them at different or at every body or every part or every tissue of our body. We can come and take a cell here and inject and insect it into our brain cells, and then that cell will produce those cells that are causing that brain damage, right? Hence some people don't want this stem cell to be tempered, because we are tampering with a human beings here. The moment this fertilization happen, some people they classify this as a human being. We classify that as a person, right? And it's not just it don't just happen. The fertilization of an egg and an ovum, it's very much complicated. You might find that your sperm here, there's a mutation in a chromosome number 11, and that mutation in that chromosome number, it's not providing a specific protein that will need this egg and this nucleus here to fuse. You understand? There's a lot to understand here. And then we also have to give you less. So if you came with this information from term 1, where they told you that these cells, they are the stem cells, and then these cells, they differentiate to different part of the body depending on the side of the cell that is situated, you are coming here and you're going to have a smooth cell. So we can also go to get those stem cell from a bone marrow, right? You'll remember that from the movies and what we need a bone marrow. Someone will sleep facing upward, and then they will go to their bones and drill the hole and take out the bone marrow, mgogo, that thing that we find in between the bone. Unali something here, there's a juicy thing here, brown like liver thing. That thing here on a bone marrow. So those cells, they are undivided cells. So if you can remove those cells and take them somewhere, they can be a muscle. So here's the thing. If we have a chicken leg, right, or a chicken, and then we take this stem cell and inject into that drumstick, what is going to happen on that drumstick? That drumstick is going to be huge, right? So this is what the technology that they are using to produce more food. Because you can just go and produce stem cells that are a cow and insect them into a specific, maybe like a forward leg, and then you produce steak, T-bone, the huge one, right? So we can also have them from the umbilical cord. The umbilical cord blood, the blood that is in umbilical cord, they also contain the sperm cell. right? And also this umbilical cord, they also say the same thing with the embryonic what. So the skin and the cartilage may also be used. So note about the stem cell technology. We have an embryonic stem cells. Embryonic stem cells are most versatile as they have the ability to form any tissue. That's what I was talking about, right? However, as the human embryos are killed, this is a controversial technology because we are killing a human embryos here. For you to have these cells, being injected to your body, maybe for treating something, maybe heart attack, you had a heart attack and then some cells in your heart, they stop functioning or they died. We can add these embryonic cells here so that your heart will be restored. But where are we getting those human embryos? We are killing humans, right? The moment this thing happens, it means that we are going to kill that human being for your benefit. Hence, we say that biotechnology is the use of a biological process to quantify or to add a quality into a human life, right? So adult stem cells that are much less controversial. This one, they don't controversial just means that people, they now be against it, right? So this one is a less controversial. A bone marrow has been used for a long time to treat cancers of a blood such as leukemia. Leukemia is the blood cancer. You know this one, this cancer here, but other types of stem cell treatments are constantly being explored. Some of the procedures have involved, right? So today we'll be learning cloning, right? So some of these procedures involve replacing dead cells in the heart after heart attack. That was I was saying. Where you have a heart attack, you can do this, or you can grow skin tissues to treat burn victim. Let's like maybe you were burned, and then when you know that when you get burned, those skins there, they are going to die, right? They are going to be cooked, and then after cooked, those proteins, they are going to be denatured. You did this during protein synthesis, and then they are going to die. After death, we can take the stem cells and come and insert them here, or we can take a skin and then insect in a lab, and then we produce a skin, and then we do a skin implant here, and then you have your new skin. And then, or we can grow nerve cells to treat spinal cord injuries and a Parkinson disease. Spine cord injuries, you are going to do a response to the environment, right? Where we are dealing with the nerve cells. And nerve cells, you understand, impulse. We are going to learn this next, right? So these stem cells, they can be used now to replace these nerve cells. If you don't know about what is a nerve cell, don't worry, you're going to learn about it when we reach that chapter. However, a great deal more research is needed before these procedures are performed. Parents who believe that they will be a success in future are able to collect umbilical cord blood from their babies at birth. The blood can now be frozen and stored for a future use. Although such facilities are available in South Africa, it is an expensive option. So it is an expensive option, but if we can look it in a business-minded person, we can say it's very much simple. We just have fridge. Then when you are born, we take your baby, we remove, or we just take the umbilical cord as it is, or we remove the blood of umbilical cord, and then we store them in a refrigerator, and we make sure that that refrigerator works until why. And then when you pay a monthly fee for us to keep your blood, your umbilical cord blood. So they say it's very much expensive option. Like, where, how? Because we don't see these refrigerators. Now we have we have what, solar load shedding. So it means that we'll have solar energy, and then we have batteries to back up these fridges, and we'll have a fridge somewhere. That in this fridge we have like a mortuary where we have a specific group, and then in this group we have your umbilical cord blood. And when someone in future is decided now, or maybe your baby now, she's having a problem somewhere, someone can come now and take this umbilical cord, and then with that umbilical cord we can go and take the stem cell, and we make them to undergo mitosis. We put them somewhere, we process them to undergo mitosis. Undergo mitosis, they divide, they become many. We take those stem cells and we inject where we want to treat the specific disease, and that disease is cured. So you see, let's move on. What very expensive, but let's move on. Then we have a cloning. But cloning, what is cloning? Simple, forward, straightforward. Cloning is the natural or cloning is a natural or let me change color here so that our concentration will be shift to something new, right? And then we said the cloning is the natural or artificial process of creating a genetically identical copy of an organism or biological material, like a tissue. Yes, those who understand things, they understand what is they're talking about. But Mr. Mpepu, can you please say something in this definition? In this definition, what we are saying about cloning, we are saying that this is a natural or artificial. I told you that natural, it's happening naturally. Artificial, it's when as human being choose that we want this specific animal and this specific animal, or we want to produce this specific animal, or we want to produce this specific artificially. Natural is not involved. Naturally, natural is involved. When a cow and cow meet, that is happening naturally. But we go and take another cow and take another cow and force them to mate. That is artificial, right? So this is the process, right? A cloning, this is the process of creating a genetical identical copy, right? When we say a genetical identical copy, we are saying that the DNA that we have in this mother, it's copy of what this offspring is having. But if we add another copy from the father here, the offspring here, he or she is going to have a non-identical copy from the mother. So you know what is copying. Copying, we take a paper, like what we take a paper, we put it in a copying machine and we produce another paper exactly with written same thing, right? But if you change the font and what, that now is not a copy. Hence, you also have to go to the police station and you certify that copy to someone to see if whatever is written here is the same as this. So that is the copy. Then here we are producing a genetically identical copy of organism, right, or a biological tissue. Like for an example, if I'm saying a liver tissue, right, so we are making more livers in a lab, right? We are using a DNA in this liver and then we add it somewhere, then we cultivate this, and then now we produce more livers. Or we just have a liver that is dividing, growing, like just but what we call it what cancer, right? And dividing what then when this liver is dividing, you just come and cut it and you take it and you sell it. Then this liver grows, you cut it, you go and sell it. We can do it in this cloning process, right? Or we can just have, for an example, I want to come, we will also have to come and explain this dolly thing and what. For another example here, before we can dive in, using the using this process to create a genetically identifiable copy, right? We can come and take one chromosome from the father. This is what this thing is saying. We can come and take a chromosome from the father and we produce a baby or an offspring that have these 44 chromosomes. So it means that this father here only 43 chromosomes, the offspring will have the exact 43 chromosomes. So we can come and in a somatic, this is what they're explaining. They say you can go to a somatic cell, since we know that our somatic cell, they have a nucleus, and this nucleus, they have 46 chromosomes. We can come and take this 46 chromosome and insert it into an ovum. In this ovum, we come and remove the nucleus, right? We remove it, and then after remove it, let's use another color here. Also, we remove this nucleus, right? We remove this nucleus here. We remove this nucleus here, and then we come to that removed ovum here, and we insert that this nucleus here with 46 chromosome. With 46 chromosome. So it means that this cell alone is going to have a same cell. Actually, the somatic cells will have many cells like this. So we just remove one cell here. And then it means that this cell, because this cell undergo mitosis, this cell here is having 100% DNA structure as this and this, right? So with this 46 here, you can come and insert here. And theoretically, what they're saying is that this cell here, this ovum, they can induce an electric shock, so that this cell here will undergo mitosis. After undergoing mitosis, it's going to form a blastocyte, and that blastocyte or that embryo, they are going to insect it inside a sheep, and then that sheep is going to produce that baby sheep called Dolly, that baby sheep with same characteristics of this huge chromosome, huge animal here, because of the small of the one chromosome that they instructed from this big what. So this process, this process, guys, I don't tshwarela. Nna ke tshepe ye angiyithembi, like, don't trust this process, guys. Le tlo ntshwarela. This one, maybe, okay, let's explain this thing, people. Maybe someone will come and change my, just to make it very much understandable to me, or this thing is possible, right? So here's the thing. But cloning happens naturally, right? Cloning happens naturally. So to them, okay, let's see. When asexual reproduction take place, you know that a bacteria will divide and form two. That is an asexual reproduction. So this cell and this cell, because they are identical, you see, remember during a recombinant DNA, the DNA here was same as DNA. Because this thing, they're identically, we refer this as a cloning, right? So another word of cloning is producing an exact copy of a something. When you have an exact copy of something, it's when we have cloned you. So it means, like, as I am now, if I'm saying that I'm cloning myself, or Kanye West bam clonile, or mang bam clonile, I will have a baby, or I will have an offspring, or there's going to produce someone that person is identically to me. So here's the thing. When you remove my 46 chromosomes and you add somewhere so that you reproduce me here, do you think, like, these chromosomes of mine that I have, how are they going to now start from the baby up until they produce something like me? So you see, this chromosome thing, yeah, I'm not sure if I will explain this so that you'll understand, but this chromosome thing, yeah, there's something, yeah. So let's learn what they're saying. Like, we are learning, and then or a plant self-pollination, you know that the plant, they self-pollinate. This plant can add, a plant like this can remove antennas here and add into another flower at the apical, and then this flower here will produce babies that are made of spermatozoa from the father and ovum from the mother. Same tree, sex. This one is self-pollination, right? And when identical twins are formed from a single zygote, so you know that with a single zygote, the blastocytes, or the mutation, that the mitosis can happen, and then one blastocyte, one egg result into two cells that are divided like this, right? So it's when the single cell zygote become two. This organism here, it's having a identical DNA as this one, right? Hence we have identical twins. This is what they are happening, right, with identical twins. So okay, so another thing, okay, let's move on. These processes all give rise to an individual with DNA identical to of the parent, where biotechnology has enabled cloning to produce a new individual that is an exact copy. This biotechnology has enabled cloning to produce a new individual that is an exact copy of the original from which the body cell was taken, right? So we take a body cell, and then the organism that is going to be produced is going to be identical. It means that it will be like this. Sorry to explain it will be like this. If now I'm like this and I'm cloning myself, the possibilities or the chances that I can have me is to have a mini me growing with similarities like me, because now I will be 49 years and this baby will be, now maybe let's say for now I'm 20 years, this baby here, she's 0 years, and by the time I'll be 49, this baby will be 29. So by the time the 29 me, I'll take photograph of 29 me, and we are going to compare with 29 that cloned thing. If this cloning can happen, this is what they are saying. Actually, they are saying it produced an exact copy of an organism from which the body cell was taken, right? It's not like they are removing a body cell, an eye, a small ear, sorry, a small piece of a cell in an ear, and then they are going to place it somewhere, and then tomorrow we wake up with a huge ear that is similar to mine. This is not what is happening. This is the process that will take time also. So let's hear what they're saying. But in July 1996, guy, this thing, this Gene Watwat, the father of Gene Watwat, it was the guy who started this 1950 something. So it took them 50 years or what to successfully clone a doll, to clone a sheep. But in July 1996, Dolly the sheep was the first cloned mammal using an adult cell. In this case, a mammary gland cell, right? So they use a mammary gland cell. You know the mammary gland cell. These are the cell that we find on a mammary gland, like they produce milk and what, right? So the what? The our breast, we have mammary gland cell, right? So this is what they happened in 1996, and they said they tried 277 times, right? They had 277 try until successfully in 1996 they cloned the first sheep called Dolly. Right? So okay, again, Jiggy in April 2023, in 2003, now, Footy, this is the cow, called Footy, the cow, was the first cloned animal in South Africa. And in this case, a cell from the ear of the prize-winning dairy cow was used. So here's the thing. We go to the ear, and that's what I was thinking. We go to the ear and we remove a cell. It's a cell, it's a microscopic thing, my guy. If for now you go to the piercing, and after piercing they remove that a small skin there. That small skin there, you might find that it's having lots and lots of cells. So you can go and take that cell in this footy guy here. You can go and take that cell. You can see this guy one. Okay, but you can go and take the cell here and then infuse it. You see the process. Let's go to the process of how this cloning, they say, is taking place, so that you'll understand now, right? How are this cloning taking place? But the ARV, that's what I was talking about, the IVF, in vitro fertilization, is not. But okay, in vitro fertilization, that thing that I was talking about, where we go to a sperm cell and then ovum, we instruct the whole body, the whole cell, and then we insert it inside the ovum, right? So what is happening here, the sperm cell, a sperm cell is like this, right? And then this portion here, it's what that houses the nucleus, and then we have the mitochondria. So we come and cut here. I've explained this. So this head is the one that we take with its full context, is that the word, with its full composition, and we insert it inside the egg. So whatever is inside here, produced by a sperm, produced in the process of spermatozoa, it's required to come and fertilize an egg. Because you know that the moment this sperm egg enters the egg wall shuts, right? That's what they told us. The moment this egg here, or the moment this content, this thing that is housing, that is going with this chromosome, the moment it enters the egg is going to produce a layer that it will not allow another cells to penetrate. So you can see that there's a chemicals and there's some components that this sperm cell have. It's not a matter of just removing a 23 chromosomes. That's what I'm saying. It's not a matter of just removing a 23 chromosome. It's not a matter of just removing a nucleus and on its own and insert it somewhere, and then we expect that this nucleus, because it's 23, and here we also have a 23 chromosomes, when this chromosome meet, they are going to mitosis and called a blastocytes. Right, okay, but the IVF, this process now, it's not cloning. Right, we say this is not cloning because as a sperm and an egg are used to form an embryo, here we use an entire egg and entire sperm to form an embryo. The embryo is genetically different to either the parents. Why? Because now this 23 chromosome, we have 23 chromosome here from the sperm, 23 chromosome, and the 23 chromosome from the mother. So with these two chromosomes, it means that we are going to have 46 chromosomes that are different from 46 chromosome of the mother and 46 chromosome of the father. Hence we say this process is not a cloning. But if we have this 46 chromosome from the father and they're sick exactly with the 46 chromosome from the daughter, yes, we say now this is a cloning, identical, right? Hence we say this IVF is not a cloning. So let's move on. And then they start with an advantage of this cloning. They said this cloning in early advantage. And then what Therapeutic cloning can replace damaged tissue, e.g. skin, heart cells, and a bone marrow, so helping to save humans' life, right? So we can do a cloning to clone those IVF or those stem cell. You'll see why this is happening. You can go to a stem cell and, you understand, clone them to produce two stem cells that we can use to treat skin, heart cells, and what. And then number B, they said genetic diseases could be prevented. And then the superior animals may be bred to improve food supply. So if we are successfully producing a superior animal, for an example, we have a big cow, then we take that cow, we do the process here, we remove its like what they did with Futi. They said Futi, it was a cow that produced more milk, right? So they came there and take a nucleus somewhere inside into the ear, and then insert it into an egg, and then that egg undergo reproduction and mitosis, fertilization, and then was implanted somewhere, and then that small animal grew up, and then that small animal was produced, and that small Futi cow, now it has a gene to produce more milk. Theoretically, it's going to produce more milk, right? So we can use these superior animals to breed and improve food supply, right? This is the benefit of cloning. And then number D, they say the research in any form improves the skills and will open the other avenues due to the spin of technology which could help the mankind in the future. So the cloning, because we are doing the researches and we have a spin-off technology, we are introducing new technology, we can now incorporate this thing to help humankind in a future, right? So let's come to the process now. This is what I want. After here we are done, right? After this process we are done with process of cloning, and then now you can go and tell me if this can be successful, this can happen successfully, right? So what is happening, the description of the cloning process that follows is related to figure of the 15. So what is happening, this is the figure 15, the cloning of a sheep. You can see here, this is what is happening. They said that, you can see, yes, what is happening. They said that a sheep A, it's a female sheep, right? This is a female sheep. You go to the sheep A, and then you go to the body cell. It's a somatic cell, any cell, right? Taken from the sheep, any cell, but in a dolly manner, but they took a mammary gland tissue, mammary gland cell, right? In a dolly what this is the thing, but you can go and take any issue this way. Yes, the mammary gland cell, right? In this case here, dolly. And then you can just go and take any tissue somewhere, because here with this tissue we just need its nucleus or its DNA components, right? So after taking this DNA components, okay, now we'll have a cell there, then we have a nucleus, we remove the nucleus, and then here's the nucleus, right? And then from this side again we go to another sheep, and this sheep is undergoing ovulation, and we take the egg cell taken from a sheep B. This sheep B here is going under ovulation, and then we take its egg, and then after taking its egg we remove its nucleus, right? This nucleus, we remove it, and then when the nucleus is removed here, the nucleus is removed, now we're just having an egg without a nucleus, and then we come and add that nucleus that we remove from this somatic cell into this egg. Theoretically this will happen kabubona when they are thinking. Hence they will undergo 227. I'm not saying this is wrong, but hence they undergo 227 failing, right? 227 times failing, because this process here, it also comes with very much questions. They remove a nucleus here, we just have a cytoplasm and whatever composition there, and then we add the nucleus of this somatic cell, because they think that this here is having 46 chromosome, right? And then the nucleus here is having 23 chromosome. So this nucleus here, it has to have another 23 chromosome from the father and make it 46 chromosome. These 46 chromosome, they are the chromosome that they are going to function, or they are going to send a message to this cell here to undergo a mitosis for it to form a morula and for it to come a blastocyte. But bona no. Because we only need 43 chromosome here, we can just go we can just go and take this 43 chromosome and add it here. Because this cell now is having 46 chromosome, it means that this cell here can now undergo mitosis. Maybe it can undergo mitosis, but that mitosis, is it going is it going to form something called a blastocyte or to form an embryo? Maybe or maybe not, but this is what is happening. After fused, the cell fuse develop into an embryo which is placed into the uterus. So in their theoreticalist theme, what they are saying is that they took this 46 chromosome and inject them inside this chromosome here, and then this inside this cytoplasm here, and then this cytoplasm, the DNA from the sheep A fused with a with an egg cell from a sheep B. So this egg is going to undergo mitosis, and then it's going to undergo the blastocytes, and then undergo and form an embryo. And this embryo, it can now go and inserted inside the sheep C, which is I said this is a what? It's a surrogate, right? It's a host. The surrogate, or it's a host that this implantation is going to take place, and then that implantation, this and this, they are not biological parents, and then the C is going to have an A, and then the A is going to be born as a lamb, and it's a clone sheep A. Why they are saying it's a clone sheep A? It means that this lamb is made up of 46 chromosomes. We come to this body and remove a somatic cell. You will see that in this somatic cell, this 46 chromosome, they will be equal to the 46 chromosome that will take from number A somatic cells. These chromosomes, they will be identical. That's what they are saying, right? So that's the process, guys. So you will explain, you will tell me on the comment section if you think this process, it can be successful or not. And then here in the explanation on our left hand side, they say the sheep A is the superior animal, right? Superior animal. They're just using some words. Superior animal is the first animal, right, to be cloned. This is the animal that we need to clone. Sheep A is the animal that we need to clone, right? We want to produce a baby that will look exactly as a sheep A. We are not going to say we have sheep A, then boom, tomorrow we have another sheep A, and then this now we have twins. No. That one now it's going to be impossible, because when this sheep grows, this sheep here will be a baby. So when we clone something, maybe for an example, a process of cloning today, we can say that we find Nelson Mandela living, a guy named Nelson Mandela now, right? Or we find a guy named those people who died back there living now. We can now conclude, this one is a clone. But for you seeing Mr. Mpepu working somewhere and say that's the clone of Mr. Mpepu, we will have to question your understanding in this cloning thing, right? So okay, this is the to be cloned, and then sheep B is the inferior. This one the superior, this one is an inferior, the superior on the top, inferior bottom, but hardly so it can survive the harvesting of egg, right? So it means that this sheep here, they can come and harvest their egg, and then this sheep will survive, right? This sheep here, they can go to their fallopian tube, or they can go to the ovaries itself, and before the process of before the process of ovulation, we take that egg, and then this sheep will survive. And then after taking this egg, we take it to the lab now, and then an egg cell from sheep B is taken, and then the nucleus is removed. We remove the nucleus. When they say a nucleus, it means that we are going to remove a body like this inside the nucleus. We are going to remove everything here. They are not just removing the DNA. They are not just removing the chromosomes here. They are removing the nucleus with its components. Aras. Then number C, a body cell or a somatic cell from the genetically superior sheep A is collected, right? When they say somatic cell, the body cell is also called a somatic cell. In this case, they took a mammalian cell, right? But in case of Futi, here in South Africa, Futi, they took an ear cell to show or to prove that this process can happen even using any somatic cell, right? The nucleus of the DNA from cell A is removed and placed into the empty egg cell, right? So we remove this nucleus, we replace it with another nucleus, and then this egg here. Here's another thing again. This egg here, instead of having two nucleus, one from the sperm cell and one from the mother, this egg cell here, jiggy jiggy, is going to have one nucleus, and this nucleus is going to provide everything that is needed for this cell to undergo mitosis and produce an embryo. This is where I'm saying something called neshayamanzi la. Something called neshayamanzi. It's either this process was never successful, or we can go to the Footy and ask them, since they are claiming that also in South Africa we had a successful scenario of this theoretically but theoretically, this is the critical thinking that they want you guys to think, right? So let's think this deeply. Let's discuss this. Do you think this is the book is telling us, but where now with your understanding of genetic engineering, do you think this process was successful? And I'm still saying no. I stand to be corrected. That is just my opinion, guys. That's me and my thinking. Maybe my thinking is short, but if you can agree with me, guys, comment on the comment section why you think about this. Ne? And then the egg cell, it's stimulated with a shock so that it starts to dividing by a mitosis. So you are going to ask yourself, but this mitosis, Mr. Mpepo, whatever you're explaining here, it seems like how is this ovum now it's going to undergo mitosis while we don't have the thing that has to come with the nucleus of a male, both the centrosomes? It doesn't even have a centrosome, what, but we expect this egg to undergo mitosis. They are saying it's stimulated electronically so that it undergo mitosis. While it undergo mitosis, we still have the centrosomes that they will have to pull this chromosome to the different side. But since we only use this 46 chromosome somehow, it compromises this experiment. Where is the ovum? Because we learned that ovum doesn't have centrosomes. Where are they going to get the centrosomes to divide these chromosomes? That's another argument, right? I still stand to be corrected. So let's move on. And then the egg cell now has a DNA from the superior sheep A, and it will grow into an embryo. Then embryo will grow into the sheep, and that sheep, it will be the identical composition of that animal there. But the embryo is then placed into the uterus of a surrogate or a foster mother. There's the word surrogate, or the foster mother, sheep C, and will develop into a full 10, right? So the baby lamb is cloned of sheep A, meaning it will be an exact copy of sheep A. So the question that they can ask you here in our question paper is that outline or briefly outline the process of cloning, right? Tell us what is happening in the process of cloning. Theoretically, you should be able to explain this, whether you understand or whether you agree with me that this process, it cannot be successful. You have to explain the entire process, right? Starting with the one cell taken from the mother, and the other cell ovum this side taken from the donor, and then we remove the nucleus, we change it, we swap the nucleus, this one we throw it away, then we take nucleus this side, we edit this side, and then this one with a 46 chromosome is going to, that's what you are going to say, this one with 46 chromosome or genetically makeup of the sheep A is going to undergo mitosis, and then undergo mitosis, it's going to form an embryo, or it's going to form a new sheep called a clone. That's what you should say, regardless of Mr. Mpepo agree if this is going to be successful or not, right? I hope you understand that. And then after that, you'll say the baby lamb is clone of sheep A, and meaning it will be an exact copy. Then they can also ask you another thing. The one that you have to know is this sheep, the first sheep is called Dolly, and the first cow is called Footy. So they can ask you this, name the first successful story, or which animal was named, or what was the name of the first animal that was successfully cloned here in South Africa? It's Footy. And which animal was the first one to be cloned in 1996? It was Dolly. So Dolly, she was the first one to be cloned, and then Footy is the second one here in South Africa. So this is what they can ask you. Maybe we still have another animals out there that they are cloning. We are not sure. But in this scenario of ours, or in this textbook of ours, they are telling us that you should concentrate on Dolly 1996 and Footy 2003. Right? So it can come in a one-word question, where they will ask you in a one-word question, it was an animal that was successfully cloned in a year 1996 July. And then when you choose, they will maybe say Dolly and Footy, or maybe they'll say this is a successful story, or this was an animal that was successfully cloned, right, that was successfully cloned. And then they will say Dolly, and then they will say Footy, and then maybe they will say Sporty. Then when you say A and B, since you know that Dolly and Footy, they are the only one that you are considered. Whether Spooky was somewhere, you read it somewhere that Spooky was also a successful scenario of a cloning. Here you don't include it, right? In our syllabus, we are only dealing with Dolly in 1996, and in 2003, South Africa, Footy. Right. So now you can come and comment whether you think this process here, it can be successfully done or it cannot be successfully done. Then the disadvantage, they are not going to ask you about disadvantage. Maybe if they can ask you the disadvantage of this is that this thing is expensive. Just stick with the word. The process is expensive. Where are you going to get a material to harvest the egg? After harvesting the egg, the moment you take it out, you have to incubate, you have to put it somewhere where it's not going to dry. You understand? Maybe by that time we have a fallopian tube. That fallopian tube, it has some chemicals that makes this egg to stay alive. When you remove it, that egg dies. So it's very expensive. That's the disadvantage, right? And then advantage, we can produce more animals. We can have animals that have a desired gene. And I understand, we can produce animal that we want to have in the near future. So you get me? So it means that disadvantage is that this thing will take time. For you to have a successful clone, you are going to have a long of time. Do you understand? For you to see this clone, if we are saying this is an animal successfully cloned, excuse, maybe for example we're having a cow here, this cow produce more milk, and then we clone it, we have a small cow there, Footy. This process, we are going to wait for a long time up until this Footy here produce another milk. So in the process, it means that you understand, we can cap or we can increase the food production, but they will have a waiting period, right? So with that being said, now I'm not sure with now if this cloning what can be successful or not. It's up to you. You can go and do the research, come back here and let's argue this one, right? And then activity 8, we'll do this tomorrow. I'll come with question papers that are related to this cloning and what, and then we do this as a class tomorrow. And then from there we can also incorporate a profile, DNA profile, and what, right? So with that being said, thank you guys. And then this the activity, go and do the activity. After activity, we will come back tomorrow, Thursday. We are going to do mitochondrial DNA and tracing. This is a small part, 32 seconds, 2 minutes will be done. And then after that we do a revision, and then Friday we do our end topic activity, this activity here, right? This activity, a genetic and inheritance end of topic exercise. We are going to do this on Friday. So tell your friends to tell their friends to come and subscribe, guys. 1 hour, 2 hours. Sorry for keeping these 2. You understand that we also have to cover the previous topic yesterday. And then today we are moving on with our knowledge base. And then after that we do activity tomorrow, Wednesday. Then after activity we do the mitochondria. You see that just only an explanation of you to understand what is happening using this mitochondrial DNA to trace a specific people around the world, right? So with that being said, thank you guys. 1 hour, 2 hours of non-stop. So guys, we can record this up until 5 hours of non-stop, but I also think of your time, guys, since you guys you still have to understand, to study something else. So that was it from me, guys. Don't forget to subscribe. After subscribing, don't forget to hit that thumbnail, that even if my teaching diabora, just hit that like button, and then after that subscribe, guys. After that, subscribe. I should write in red. Subscribe. Right? And then after subscribing, hit that notification bell. This bell will be like this. Hit that notification bell. Right? Hit that bell. Right? So that it will be notified when I send a new videos. Right? Another thing, don't forget to comment. You can just comment and share the link. Don't forget to share the link. Right? Share the link. Let's share this. Don't forget to share, guys. Just share this content with other students. Right? With other students. So thank you, guys. See you tomorrow. And for those who are here, hey, I don't know, Zama, guys, let's just study this together and let's move on. Thank you for being here. You can see this are knowledge based a knowledge based topic. So you'll have to understand this thoroughly so that you'll be able to explain this to someone else. Right? When you know this, tomorrow you know how to explain this. After a year, you can say, now I want to become a genetic modifier. I want to do this thing at cloning. So you'll understand that you'll have to work based on life science after obtaining some marks. Hence, this one, it contribute 50%, right? Out of 150. So it means that 50, if you have out of 150, if you get this 50%, when we have a positive feedback, yeah, this person understood this part. So that way you can now go and apply for something that to the universities that they will deal with this. Now you are applying, right? Now we are applying. It's not only medicine, it's not only a dentist, it's not only optimists, it's not, you understand, you can also go there. You can also go and apply for scholarships to go and study this information abroad. You go UK, go USA, we have scholarships for those. And then after that, you study in those universities, you come back, and then you open your fridge company that I was talking about, and then you produce these things. Right? You have a certificate, because as like now like this, I can just go to a DNA and remove DNA and produce something. They are going to arrest me because I don't have a certificate. Right? So with that being said, guys, our 2 hours, it's up. Thank you, and thank you for being here, guys. Thank you for tuning in. Thank you for watching my videos. So for those of you who haven't subscribed, guys, can I please ask you for the last time, can you please subscribe? Right? Or press the center thing here. Press that center thing and share the link. That is from me, guys. Thank you, Mr. Mpepu. Fine. See you tomorrow. Sharp. Peace be unto you. Ukurula kube nani. Khotsong ayebelilina. Ishe.